Welcome to the second module for Prime Immunology. In the previous module, we talked about the cells and basic functions of the innate immune system. Throughout this module, we will address key concepts of adaptive immunity. The adaptive immune system, as its name suggests, is defined by its specificity and thus a delayed reaction. This is the arm of immunity that vaccines take advantage of. It has a memory and can therefore better protect from future infection by the same agents. The key theme in adaptive immunity is self versus non-self. That is, adaptive immune cells hold the ability to differentiate between cells belonging to the host versus those belonging to foreign bodies. Unlike innate immune receptors such as TLRs, which specifically recognize conserved molecular patterns found across microbial species, adaptive immune cells instead have been primed through an extremely intricate and finely tuned process to tolerate host cells and attack foreign ones. In this way, they are able to distinguish that which is self from anything that is not. The two classes of lymphocytes that make up the adaptive immune system are T cells and B cells. B cells differentiate in the bone marrow and then travel to lymphoid organs. T cells instead differentiate in the thymus, but also migrate to lymphoid organs. An easy way to remember this is T for thymus and B for bone marrow. As a historical fun factoid, the B in B cell does not actually stand for bone marrow. Instead, it refers to the name of the place where these cells were discovered, that is, the bursa of Fabricus in birds. T and B cells not only differentiate separately, but also play roles in two separate branches of adaptive immunity. The arm of adaptive immunity orchestrated by T cells is known as cell-mediated immunity, while that involving B cells instead constitutes humoral immunity. Ultimately, cell-mediated immunity targets infected host cells and therefore plays a crucial role in fighting off intracellular infection. Humoral immunity is instead more effective against extracellular microbes. These two branches, however, do talk to each other and are often co-involved. T cells undergo an extremely complex process of differentiation that includes both positive and negative selection. You will learn the details of these processes in your immunology block. Ultimately, the end of the maturation process leaves us with active or effector T cells. For simplicity, we are going to talk about two classes of effector T cells, which are defined by the surface markers expressed on the T cell itself. Thymocytes that are positive for CD4 are known as helper T cells, while those positive for CD8 are known as cytotoxic T cells. Helper T cells, as their name suggests, play a supporting role. That is, they direct other T cells, B cells, and phagocytes by secreting chemical mediators to orchestrate a potent immune response. Cytotoxic T cells instead directly kill any cell that has been identified as infected. Do you remember which innate immune cell had a similar function to the cytotoxic T cell? One of the many interesting things about T cells is that they are not able to recognize antigens that are floating around in circulation. Instead, they require fragments of foreign agents to be displayed by specialized MHC molecules on the surface of antigen-presenting cells. MHC stands for Major Histocompatibility Complex, which we touched on slightly in the innate immunity module. MHC type 1 molecules are found on all nucleated cell types in the human body and present peptides from antigens that are found in a cell's cytoplasm. MHC1 molecules are recognized by CD8 or cytotoxic T cells. MHC2 molecules are instead found only on specific types of antigen-presenting cells, dendritic cells, macrophages, and B cells. They present peptides from antigens found within a cell's vesicles following phagocytosis, and are recognized by CD4 or helper T cells. Moving away from T cells, the humoral branch of adaptive immunity is initiated when a naive B cell recognizes antigen. This cell then undergoes a process of clonal expansion and differentiation to then mature into a plasma cell that secretes antibodies specific to the antigen target in question. When an infection has been cleared, a pool of so-called memory B cells remains behind, so that any subsequent invasion by the same pathogen can be fought off immediately. Antibodies themselves function in several ways to fight off infection, including neutralization of microbes, opsonization, complement activation, phagocytosis, and more. In the next slide, we will discuss key functions of different antibody types, and I will cover opsonization and complement activation in the third and final module. Specific types of antibodies will predominate during a given infection, depending on the timing and/or particular agent in question. Immunoglobulin G, for example, functions to neutralize invading microbes, activate complement, direct natural killer cytotoxicity, and provide immunity to neonates through transfer via the mother's placenta and gut. IgM is instead one of the first antibodies to form, around 2 weeks following an initial insult, and, like IgG, acts to activate complement. IgA is the critical antibody for mucosal immunity, via its secretion into the lumens of gastrointestinal and respiratory tracts. And IgE plays a key role in protection against helminths, or worms, and in allergic or immediate type hypersensitivity reactions. You will cover these antibodies again in your immunology block. This slide provides a summary of what we talked about in terms of adaptive immunity. Thank you.